A-Level Biology 9700, Paper 4, Variant 2, October, November 2024, question number 5 and 6. So, question number 5 is phenotypic variations exist in natural populations. There are many causes of variations. Natural selection determines which phenotypes are advantageous. So variation is a particular characteristic can be described as either discontinuous or continuous. And the table 5.1 contains a list of statements that apply to discontinuous variations. So continuous variations are both complete, both columns of table 5.1, put a tick in the box if the statement applies and leave the box empty if the statement does not apply. So here the students who know the concept of discontinuous and continuous variations, they can easily solve this question. They can fill up this table. So first of all, the natural selection. Natural selection is a phenomena by which we say that the nature select the organisms which are best adapted to the environment. So in this way, we can say first of all we have to discuss which one the differences between continuous and discontinuous variations. So first of all, I would like to say the continuous variations are those types of genetic variations which shows and you can say a range of phenotypes of a particular characteristic in the population. The continuous, and we will write this continuous on that side. So continuous variations show the range of phenotypes. So it means the variations are originated in every generation. So whereas in discontinuous variations, they show separate forms of characteristics in the population. So they are limited. They have discontinuous range of phenotypes. Or we can say one or only few phenotypes are available. So in case of you can say continuous variations, the intermediate groupings are present. And whereas in discontinuous variations, intermediate grouping absent. And next we can say the more common type of genetic variations are there. So many genes are present for the determination of a particular trait. Many genes are involved. One or few genes are involved. So examples we can see, for example, weight, height, because the weight and height, length of organisms, all they are changed in every generation, or within the same generation there are variations among different children or of or organisms. Whereas in discontinuous variations, there is you can see, we can give an example of blood groups. For example, blood groups, and which are A, B, and O. There is no other type of blood group available. Only a limited 4 types of blood groups are available over here. So in the light of all these, we are going to this open involves one gene only. So this statement is correct only for discontinuous variations, as already I have written over there. Only one gene is available, or this is for discontinuous variations. Next, environmental factors may affect gene expression. So in discontinuous variations, as AB, O blood groups are example, so there is no environmental factor will affect the blood group. As for example, if mother and father, they have blood group A, B, so the child will have the blood group according to those combinations which will occur during the process. But the continuous during the continuous variations, their environmental factors are also involved. So we will put a tick over here. So there is an additive effect of genes that contribute to the phenotypes. No additive effect will be involved. No environmental factor will be affected. So only continuous variations are produced. They can involve an additive effect of genes. So in this way, there will be the range of phenotypes, as already I have told you that. So next is there are distinct difference between the various forms of a characteristics. So we can say that there are distinct differences between the various forms of a characteristic. So these are the difference. So that is the we can say the distinct differences are between the various forms of characteristics. So they are so that is the reason they are called as the discontinuous variations. As discontinuous variations, they are not changed in every generation. They are limited to a range, whereas the continuous variations have not a range. It is unlimited range of the genotypes or phenotypes. So in this way, the students who have know the properly the definition or complete information about continuous and discontinuous variations, they are able to solve this question very easily. Now, next question is, there is a variation in the quantity of vitamin D stored in the body. So vitamin D has an important role in keeping bones healthy. The main storage form of vitamin D in the body is serum 25 hydroxy vitamin D. A study was carried out on 262 healthy women to investigate if the concentration of serum 25 OHD varied between summer and winter. The women had taken no vitamin D supplements. The range of the women in the sample was 40 to 72 years old. So the range of the age was 40 to 72. So the table 5.2 shows the results of the study. You can see the sample during summer is N, the value N, 138. So the sample during winter, it is N is equal to 124. So N1 is 138, N2 is 124. So whole sample is total of this, that is 262. So the mean concentration of serum 25 OHD is 32.7, 28.5, and the standard deviation is 7.6 and 8.3. So in this way, they have given us the different values and of the concentration. So additional analysis shows that there was no significant correlation between age and serum 25 OHD concentration. So here I would like to say, explain what is meant by standard deviation. So standard deviation, students, this is a statistical measure that quantifies the amount of dispersion or spread of a set of data values around their mean. So how much the data is spreaded around the mean value. So in this way, we can say that in simple terms, it tells you how spread out the individual data points are around the mean or average of the data set. I think I would like to write this answer as well. So in simple terms, it tells you, it tells you how spread out the individual data points are around the mean or average of the data set. So I would like to say that it shows or it suggests a greater degree of variability or inconsistency within the data. So if the high standard values are there, a high standard deviation indicates that the data points are generally spread far away from the mean. And on the other hand, low standard deviation indicates that the data points tend to be clustered closely around the mean. So actually, it shows the distance. So zero standard deviation, it will show zero means that all the data points in the set are identical to the mean. So there is no variability. The high standard deviation shows the high variability, low standard deviation shows the low variability. When there is zero standard deviation, it means there is no variability. So this is the you can say this in summary we can say that the standard deviation provides valuable standard deviation provides valuable information about the consistency and variability within our data set. So a smaller standard deviation suggests more homogeneity, and while a larger standard deviation suggests more heterogeneity among the data points, and zero will show no change, no variability. Now we are moving towards the next question. And the t-test was used to compare the mean values, mean concentrations of serum 25-OHT when sampled during the summer with the mean concentration of serum 25-OHT when sampled during the winter as shown in table 5.2. So calculate the value using the formula provided. So here you can see that you have to take the values, from the table and you have to use those values in this table. You will put the values in this table. As for example, I would like to write that as for example, the x1 value, this is the mean value, and s value is the standard deviation and sample size number of observations. So I am moving towards the back. Here you can see these are the mean values, or these are the x values. It will show the x values, it will show the n values. So it is the standard deviation. So we will make use of 138, 124. This is N1 and N2. And whereas X1 and this is X2 and standard deviation 1 and this is standard deviation 2. So I would write all this as, for example, the N1 is equal to 138. The N2 is equal to 124. So X1 is equal to 32.7 and X2 is equal to 28.5. From this table, I have taken all this. S1 is equal to 7.6 and S2 is equal to 8.3. So we are making use of these values in this diagram, uh, in this formula, and in this way we will make use of all this and we will calculate the value of t. So I am going to put the values in this formula on according to this formula. T T is equal to now first of all x1 value x1 is 32.7 minus 28.5 and then and S1 square is value of S1 is 7.6 square over N1 and value of N1 is 138 and plus the value of S2 square that is 8.3. So 8.3 square divided by 124. After this, I will continue this calculation. I'm writing on screen, so it is very difficult for me to write in very small letters. So it is, I will make use of bold letters. So again, T is equal to 4.2 and divided by under root 57.76 divided by 136 plus it is 68.89 divided by 124. This is 136. Let me correct it. You know, during writing, it can happen. Okay. After I'm further continuing these values, this is 4.2 divided by under root 0.425 plus 0.555. Again, when I'm writing on screen, any problem can occur. 0.55 and 5. So after this, after it is 4.2 under root 0.98994 and at last it is equal to 2 point. Let me calculate. This is equal to, I think I should write over here. So it is 4.2 over 0. Let me calculate with calculator. Yes, it is 4 point. Answer is, the value of T is 4.25. So this is the answer. For value of T is 4.25. I have calculated in front of you. Now I am moving towards the next question. And next question is the critical value at the 0.0001 probability level is 3.77. So state the conclusion that can be made about the results of the study shown in table 5.2 and explain how the result of your calculation and B2 can be used to support this contrast. So in the results are 4.25. So the calculated value of 4.25 is greater than the critical value of 3.773 at the 0.001 probability level. You can say this value is greater than this value. We can write it 4.25 is greater than 3.773. So you can say at this probability level, actually this critical value is a point on the test statistics distribution that is compared to the test statistics calculated from the sample data. So it defines the rejection region. If the test statistic fall into this region that is more extreme than the critical value, we reject the null hypothesis. On the critical value is determined by the significance level of the test. In this case, the probability level of 0.001 can be considered as a very stringent significant level. So in this way, I would like to say that that this means the difference in serum 2,5-OH concentration between summer and winter is statistical significant because this value is larger than this value. So it shows the significance in the data. So therefore, we can conclude has a significant effect on the concentration of serum 2,5-OH. The result from previous question supports the conclusion between between because a test statistic greater than the critical value indicates a very low probability that the observed difference is due to chance. So in this case, this hypothesis, um, it provides you need the actual test statistic value from uh, which we have taken 4.25. So the critical value is used to make a conclusion in hypothesis testing, and how you calculated result would support that conclusion. So very low probability level 0.0001 signify high standard of evidence required to if we want to reject this null hypothesis. So here I would like to say that that the 4.25 is greater to the critical value. It shows that the serum 2,5-OH concentration, you can say, show the significance. So the difference, there will be a big difference between the values, statistical values, that is between summer and winter. So we can conclude that the season in this way, if they show the differences between summer and winter, so these variations help us to conclude that the season has a significant effect on the concentration of serum 25 OHD. So it will be different in summer or it will be different in winter. So this results from the result from previous, that is 4.25 as it is greater as comparison to critical value. So it also supports our answer, our conclusion. So a test statistic greater than the critical value, so very low probability that the observed difference is due to chance. So it shows due to these environmental temperature. So it shows the differences between these environmental temperature. So next is suggest likely causes the variation in quantity of vitamin D stored in the body in this sample of so. So here I would like to say that causes of variation in quantity of vitamin D stored in the body in this sample of women. This question is also related with the previous question. That is, the quantity of vitamin D stored in the body of a sample of women can be due to a uh, multiple factors. We here you can see that due to sunlight exposure, due to less sunlight exposure or great sunlight exposure, there can be the variations in the quantity of vitamin which is stored in the body. So number one factor uh, can be the sunlight exposure. As for example, you can see uh, the angle of the sun, and during the winter months or when the sun is lower in the sky, ultraviolet you can say intensity decreases, leading to reduced vitamin D production. Whereas in the you can say, so in this way the production or the storage of vitamin D can be changed or different. Secondly, the time of the day is also a factor due to which also affect the um, quantity of vitamin D. As for example, the angle of the sun affects the this intensity. So midday sun generally provides the most ultra violet radiation. So in this way, I would like to say that there can be the difference um, in the different durations of time, different timings of the day, we can say. And then skin exposure is also a factor. The amount of skin exposed to sunlight directly influences vitamin D synthesis. Cultural or clothing habits that involves covering most of the body will reduce vitamin D production. So I would like this skin exposure. If skin is more exposed, then more vitamin D will synthesis takes place, whereas the less exposure to sun. And then use of sunscreen, it also affects. Then dietary intake, vitamin D rich foods, consumption of foods which are rich in vitamin D, such as fatty fish, salmon, egg yolk, and milk, etc., can increase the vitamin D stores in the body, and a diet lacking these foods can lead to lower vitamin D levels. Number three, supplements can also increase the quantity of vitamins D stored in the body of the different persons. And then number four, we can say the age. Age is also a factor. So the capacity of the skin to synthesize vitamin D decreases with age. With the age, the vitamin D synthesis decreases as age increases. So in this way, we can say that these are there are also some genetic factors, environmental factor, genetic factors. Genetic factors are also there, which can increase or decrease the variations. Um, the individual genetic variations can influence how efficiently the body synthesizes, absorbs, or metabolizes vitamin D, and then lifestyle factors. Lifestyle also act as factor. So in this way, we can say the specific causes of variations in the sample of vitamin study would need to collect data on these variation factors and analyze their correlation and with the measured vitamin D level. So all these factors can play a role. You can write any one of these with explanation. Now it was all about question number 5. Now we are moving towards the question number 6. So DCPIP is an indicator and can be used to determine the rate of respiration of organisms such as yeast, says the category of indicators to which DCPIP belongs. So first of all, what is DCPIP? So DCPIP is dichloro phenol dichlorophenol Olein dophenol. So this is DCPIP. So it belongs to the category of redox indicators. And what are redox indicators? So these are the substance. So these are the substance that change color depending on whether they are oxidized or reduced. They change color. They change color whether they are oxidized or reduced. And their color change. Describe the color change. So their color change occur from blue to colorless. So in the presence of DCPIP, the dye can accept these electrons. So I would like to write the answer of the next question. Now, describe the color range change that occurs in DCPIP during experiments to determine the rate of respiration. So here I already told you that during respiration experiments with yeast, DCPIP is typically used in its oxidized blue form. So it is used in oxidized in oxidized form, which is blue. So as yeast respires hydrogenase enzyme, respiration yeast respires, then hydrogenase enzyme hydrogenase enzyme remove hydrogen ions. Remove hydrogen ions and electrons from transport chains. So in the presence of DCPIP, the dye can accept these electrons. In the presence of DCPIP, the dye can accept these electrons and becoming reduced. Reduced and color it changed to colorless. So I have written blue to colorless. That is when it is oxidized, it is blue. When it is deoxidized or reduced, it is colorless. And next question is, an investigation was carried out to determine the effect of temperature on the rate of respiration of yeast. And here you can see that suspension of yeast cells was added to a test tube containing glucose solution. A further 4 test tubes were set up in the same way. One test tube was placed in a water bath at 10 degrees C for 5 minutes. DCPIP was added to the test tube and the time taken for the DCPIP to change color was measured. The experiment was repeated using the other test tubes at 20 degrees C, 30, 40, and 50. The results are shown. You can see over the results. Stay two variables that need to be kept constant in this experiment. The experiment, you can see here the temperature, two variables are there, the time and temperature is there. So state two variables that need to be kept constant in this experiment, because the experiment can be conducted on 20, 30, 40 degrees centigrade and 50 degrees centigrade. So for this purpose, there should be uh, the temperature, the optimal temperature is 40 degrees. So in this way, there should be the range of temperature is there, that is 20 to 50 degrees centigrade. So we have to keep the concentration of glucose constant at every temperature, so we can find out the difference between the results of these experiments. And secondly, the concentration and volume of yeast suspension should also be kept constant for this experiment, as we have to find out the time taken for DCPIP to change color. That is, from it will change color from blue to colorless. In oxidized form, it is blue color. In reduced form, it is colorless. So during this whole experiment, there should be, we need to keep constant the concentration of glucose and the concentration and volume of this constant. Now we are moving towards the next question. Explain the results shown in figure 6.1. Figure 6.1, 6.1. First of all, I'm going to towards the figure 6.1. So you can see figure 6.1 is there. You can see on this graph the effect of temperature on the time taken for the enzyme catalase reaction of DCPIP. So you can see at temperature 10 degrees C, the more time is taking to change from DCPIP to change from blue to colorless. So then as temperature is increasing, the time to change is decreasing. You can see this is the highest point, which is at 10 degrees centigrade. So from 10 degrees centigrade, as temperature is increasing, the time is decreasing. And we know that this is the 40 degrees centigrade is the optimum temperature for enzyme activity. And you know very well you about this characteristic of enzymes that the optimum temperature for enzyme activity is 40 degree. Optimum temperature. Optimum temperature for enzyme activity is 40 degrees centigrade. Then you can see after this 40 degrees centigrade, you can see a straight line. Here you can see the lines are in different directions and temperature increasing, and then in this way the curve is not straight curve, but this you can see this curve is very straight. After 40 degrees C or till 50 degrees centigrade, the curve is very straight. So this curve shows that the enzyme has been denatured over here. It is denatured over here. So that is the reason the curve has become the very straight. So figure 6.1 demonstrate the typical effect of temperature on enzyme activity. This reaction rate increases with temperature up to an optimum point, after which further increase in temperature lead to rapid decrease in enzyme activity due to denaturation. Students, as temperature is increased, the kinetic energies also increase. But as you can say, but the optimum temperature is 40 degrees centigrade. So at this temperature, the highest activity takes place. That is, after that, as temperature is increasing, enzymes, they are protein in nature. So at high temperature, their three-dimensional structure is disrupted. They are denatured. So active sites of the enzyme change shape. So preventing the substrate DCPIP in this case from binding effectively. So thus slowing down or stopping the reaction. So DCPIP DCPIP, this is a substrate. So it will not type, so its activity or its function to blue to colorless will not take place. So it will not take place after 40 degrees centigrade. So 40 degrees centigrade is the optimum temperature for it. So it is all about the question number 6. Please like, share, and subscribe my channel. Thanks for watching.